welcome everyone to this comprehensive 30-minute video lecture on the steps involved in genetic engineering. Today we will embark on a journey to unravel the genetic puzzle and understand the intricate process of manipulating DNA. So let's dive in and explore the key steps of genetic engineering. In the last lecture we have discussed the tools that were used to manipulate and modify the genetic material. DNA needs to be identified to understand genetic engineering. In the nucleus of each organism, there are filamentous structures called chromosomes in which the genes are present. It was discovered in 1869, but its significance was only recognized in 1944. Here in this figure, we can see the nucleus present in a cell in a brown color, then the enlarged view of nucleus where we can say 24 chromosomes in a human nucleus will be present, and these chromosomes, they are condensed structure, they contain DNA, and these DNA, the functional unit of these DNA, they are basically encoded in the form of genes, or we can say they are the segment of DNA which has functions. The first DNA molecule or recombinant DNA molecule or rDNA molecule was produced in 1972 by Paul Berg by combining the DNA of the monkey virus with lambda virus, while the first genetically modified organism GMO was bacteria which was resistant to the antibiotic kanamycin, and it was created by Herbert Boyer and Stanley Cohen in the year 1973. Recombinant DNA technology, mutation, protoplast fusion, etc. are used for manipulating a gene. Large amount of knowledge and expertise is required for the same. An understanding of the whole genome sequence will also be required. It is a powerful tool for placing the multiple desirable traits in an organism which is not having it naturally. The main objective of recombinant DNA technology is to manipulate or modify the genetic material of an organism to incorporate the desirable traits. rDNA technology is the main pillar of genetic engineering or engineering the genes. There are different types of techniques which are used for genetic engineering. The first one is recombinant DNA technology, and I will discuss the steps involved in it in further slides. Then the second technique is gene delivery, that is direct introduction of the fragment of gene or gene of interest or transgene either into the nucleus of an organism or at a targeted site. By using several techniques, these techniques can be physical, chemical as well as biological. In this figure, this is an example of a physical technique of gene delivery, that is basically electroporation. From the name itself, the pores are created for insertion of the recombinant DNA molecule into the host organism. So, in the first figure we can say that before pulse the cell membrane was continuous and no pores are present. Then in presence of an electric field, from the pulse itself we can understand that it is a short influential electric field, and based on that there will be change in the charges around the cell membrane and pores are created. So, after pulse some small pores are created that will allow the entry of this recombinant DNA into the host organism, and after certain period of time these pores are transient, they will close and the host organism will retain the recombinant DNA molecule. Then this is an example of gene editing. From the name itself, just like how we edit the Word file, how we edit the photos, how we edit any videos, here it is more fascinating, we are editing a gene. So, how? Here we can see that it is a double stranded DNA molecule, and if by any chance there is a mutation. So, we will cut the DNA at the mutated site, repair the DNA where the mutation was present, and it was again reinserted. So, this process is known as gene editing, or in short it is a process of cut, copy, paste. The entire process of genetic engineering can be divided into 5 broader steps. The first one is selecting and isolating the candidate gene, selection and construction of a plasmid. Third one is gene transformation. Fourth is insertion of DNA into the host genome, and the last step is conformation of the insert. Here is a flow form of the different steps involved. Selection of gene of interest. Now, we have to select the gene which carries our desired trait. Then the second step is selection of plasmid. There are various plasmids, already I had told you in brief, but later on we will see what are the specialities of these different type of plasmids and how they can be used, or how it can be decided that which plasmid needs to be used for cloning. Then the third one is selection of a vector molecule, or we can say a vehicle which can be used for inserting the recombinant DNA molecule into the host organism. Then the next step is selection of method. Just now I told you there are physical, chemical or biological method. Depending on the host cell, depending on the conditions, depending on the size of the construct, the selection method will be decided. And the last step is how the transformants will be selected. We will discuss each of these steps in detail in the subsequent slides. The first step is to identify the specific gene or DNA sequence of interest. Let's delve into this crucial initial phase. Scientists start by researching the genetic information related to the trait they wish to modify or the protein they want to produce. Once it is identified, the gene is selected for further analysis. This gene is also known as transgene, gene of interest or foreign gene. Why it is known as foreign? Because initially it is not present in the host organism, and that is why to incorporate that particular trait in the host organism, we are inserting this foreign gene into it. Gene isolation basically involves extracting the gene of interest from the organism's DNA. Techniques like PCR helps in amplifying and obtaining sufficient quantities of the specific gene. Selection and isolating the candidate gene. The gene must contain a sequence of DNA that we want to study. Gene should have high GC content, that is guanine cytosine content should be high. Lower repetitive DNA sequence, there should be less number of repeats in the DNA sequence. The gene of interest must not be very long. The candidate gene must have a start and stop codon. Start codon means AUG is a triplet codon which has to be present in your gene of interest, and it is very essential for transcription and translation process, or in short I can say it is essential for the process of protein expression. Ultimately, the main goal of inserting this construct into the host organism is its expression, or we are interested only in the production of a protein carrying that desired trait. And the gene must also have a stop codon. There are basically 3 stop codons. They are UAA, UAG and UGA. These codons will help in stopping the process of transcription and translation at that particular DNA site. These are the characteristics which has to be present in the gene of interest. The gene of interest must be isolated or separated from the other genomic part of the DNA. This is a figure which is showing in short how the gene isolation can be done. The first step in it is the cell lysis. Then after lysing the cell, we will treat it with the protease and RNase in order to destroy the other unwanted proteins and RNA molecule. Then after that, we will precipitate all the unwanted thing in the form of pellet, and the upper clear liquid, or you can say translucent liquid, will be collected, which is termed as supernatant. Now, in this supernatant we have our DNA dissolved. So, this DNA needs to be precipitated by using some solvent extraction method, and after the precipitation of DNA we can see it in the form of coiled threads. So, the DNA is extracted in the form of threads, and then here we have the genomic DNA in our hand. So, then thereafter the restriction endonucleases will be used to cut the desired fragments from the genomic DNA to get our gene of interest. Selection and construction of plasmid is the next step. Here, vectors like plasmids are chosen as carriers to transport the isolated gene into the host cells. Plasmids are extrachromosomal circular DNA molecule. Why we are saying it as extrachromosomal? Because these are not present in chromosomes and they are not linear, they are circular DNA molecules which are first time invented or discovered in bacteria. These are used as a vector molecules because they are easy to manipulate in the laboratory. They are used as a vehicle for transferring the gene of interest to the target location in the genome. They have efficient transfer of gene at the target location. They have a selectable marker that is introduced in the plasmid DNA to identify the recombinant DNA. We will discuss what do you mean by selective marker later on. So, here is an example how the vector molecule is basically created. I told you that these are the extrachromosomal circular DNA. So, in the first figure or in the first cell we can say the plasmid is present in a circular form, and then this circularized DNA is basically isolated. After isolation, this circularized DNA nick is created with the help of the same restriction endonucleases which was used for cutting the gene of interest. Then these two will be joined with the help of ligases, and this is a recombinant DNA or rDNA which is created. It is basically created by recombining the vector molecule as well as gene of interest. Then in the next step we can see the recombinant DNA molecule is basically uptaken by the host cells. The gene of interest is inserted into the selection vector with restriction enzymes, and the vector DNA creates sticky end to facilitate the ease gene transfer. The recombinant vector now containing the gene of interest is introduced into the host cell through a process that is basically known as transformation, where the host cell takes away the foreign DNA. Here is a figure that depicts the same, and I told you that these vectors they have a selectable markers. So, these selectable markers can be in the form of a specific gene. Here in this plasmid the selectable marker was a antibiotic resistance gene. Here we can see that the restriction enzyme was used for cutting the gene of interest. The gene of interest is basically depicted as pink in color in this particular diagram. Nick was created, recombinant DNA was created, and this is the modified plasmid. Here we can see the green region that indicates the selectable marker region, and this region is basically an antibiotic resistance gene. Apart from this, it will also have an ORI site, or you can say that it is the site of origin where the replication begins. Then the next step is transformation into the host genome, that is uptake of this recombinant DNA molecule into the host. This process is known as transformation, and the cells which are capable of uptaking this recombinant DNA, they will be known as transformed cells, whereas which are not able to take, they will be termed as non-transformed cells. There are various methods for recombinant DNA insertion, and they can be either a single method or A variety of methods can be used depending on the cell types or size of the rDNA or construct molecule. The methods can be by using stress, microinjection, electroporation and many more. With the gene successfully inserted into the host cell, the next step is to culture these cells and express the desired trait or protein. We have already seen how the transformed cell will carry the recombinant DNA molecule. Then these cells needs to be cultured in either liquid media or semi-solid media depending on the conditions, depending on the cell types, and in this way we can produce large number of clones of the transformed cells or large copies of the clones of the transformed cells. Host cells containing the recombinant vector are cultured in a controlled environment such as bioreactor, where they multiply and form a cell culture. So, here we can see it in figure, the steps which we have discussed so far. The right side of the figure indicates the transformed cells carrying the recombinant DNA, and in this way we can create a large number of copies of the transformed cells. Inside the cultured host cells, the recombinant gene is transcribed into messenger RNA or mRNA and then translated into the desired protein, allowing the expression of the gene's function. After the gene has been expressed, the next step is to identify successful transformations and test the outcomes. So, not all the host cells may successfully take up the recombinant DNA. There can be, the left side indicates that there can be cell with a recombinant DNA molecule, the right side indicates there can be cell with a plasmid, and both transformed as well as non-transformed cells are present initially in the mixture. We need to separate them, and that is done with the help of culturing it in media. So, how you will separate that I will discuss in the next slide. The selected cells undergo various tests including genetic analysis and protein characterization to verify the presence and functionality of the desired gene and its protein product. Confirmation of interest. Various molecular genetic technologies are used to confirm whether the rDNA is inserted in our target host cell or not. The presence or absence of a selectable marker is used to differentiate the transformed cells from the untransformed cells. The second method is by the help of blue white colony selection, and the next method is PCR based detection method, which is most widely used or accepted. Blue white screening. The use of chromogenic substrate, or we can say colored substrate, to detect a particular enzymatic activity is the basis to screen the desired clone in blue white screening or blue white colony selection. The colorless compound X-gal or 5-bromo-4-chloro-3-indonyl beta D galactoside was used in the screening method, and it is a substrate for beta galactosidase. The enzyme beta galactosidase is the product of LAGZ gene of LAC operon. Beta galactosidase is a tetrameric protein, and an initial N-terminal region of the protein is very important for protein activity. In this system, host contains LAGZ gene without the initial region, whereas vector contains alpha peptide to complement the defect to form active enzyme. As a result, if a vector containing alpha peptide will be transformed into the host containing remaining LAGZ, the two fragments will reconstitute to form active enzymes, or we can say colored product. In addition, the alpha peptide region in the vector contains MCS or multiple cloning site, and as a result of insertion of gene fragment, consequently alpha peptide will not be synthesized to give fully active beta galactosidase. The enzyme beta galactosidase oxidize the substrate X-gal to form a product that is 5-bromo 4-chloro-indonyl and galactose. The indoxyl derivative is oxidized in air to form a blue colored dibromo dichloro derivative. Hence, blue colored colonies indicates the absence of insert, whereas colorless colonies indicate the presence of insert, and this is well shown in the figure. So, this is an example of blue white screening. The first figure indicates the non-transformed cells, second indicates transformed with non-recombinant, and the third one indicates transformed with recombinant vector. So, here the selectable marker which was present is antibiotic resistant marker. For more specifically, it is an ampicillin resistant gene, and the vector which is used is PUC8. It contains the intact LAC-Z gene. So, in the second half what we can say, this is your recombinant PUC8. Here in the LAC-Z region or MCS region of the gene, the DNA insert is basically incorporated. Then when there is no recombination, then what is happening? Beta galactosidase produces X-gal with blue colored product, and we can say that this blue colored product is the transformed with non-recombinant, whereas the other half no beta galactosidase is produced. So, X-gal cannot be splitted into the chromogenic product, and based on that we can select that the blue ones are the transformed with non-recombinants, whereas the white ones are the transformed with recombinants. So, this is the basic principle of blue white colony selection. To be more precise, what has been done here, it is combined with the selectable marker region technique, that is, this particular mixture is cultured in presence of an antibiotic. So, what will happen when it is grown in presence of antibiotic? What will happen? Only the recombinant vectors will grow, because it has an ampicillin resistant marker gene. So, the colonies which does not have recombination, which does not have recombinant vectors, will die in presence of antibiotic because they will be sensitive, whereas the vectors have specific gene for antibiotic resistance, so they will grow. So, here this is the basic selection criteria for blue white colony screening. Gene transfer using bacterial infection, chemicals in gene transfer, sonication, liposome mediated gene transfer, these are the other techniques. Gene transfer using bacterial infection basically involves deliberately infecting the host organism with bacteria, but again this bacteria is not the original bacteria, it contains our gene of interest, it is modified and our gene of interest is introduced into the bacteria, so that the host cell is infected with this modified bacteria and the gene of interest can be inserted. Chemicals can be used like PEG or other detergents can be used. Sonication, again high frequency sound waves are used. Liposomes, again it is basically a fat molecule which is double membranous, and they will fuse for inserting the transgene into the host organism. The purpose of developing the genetic engineering or genetic manipulation technique is to produce organisms or phenotypes which are useful to us. Genetic engineering techniques are used for construction of genetically modified plant species, commercially valuable organisms, therapeutic drugs, prevention of genetic abnormalities. In conclusion, the steps involved in genetic engineering paved the way for groundbreaking advancements in science and technology. As we continue to explore the potential of genetic engineering, let us always approach this field with a sense of responsibility, ensuring that we use these tools ethically and for the betterment of humanity and environment. Let us have a quick recap of this step. The first step is isolating the gene of interest. The second step is preparing the plasmid molecule. The third one is creation of recombinant DNA molecule. Then the last one is introduction of the recombinant DNA molecule into the host organism. Then thereafter clones will be produced in sufficient amount. Then what is the ultimate step, that is the screening the recombinants, screening the transformed cells and going for expression of the recombinant DNA molecules in the transformed cells. In the figure which we have seen just now, in the first step what is happening is the required gene is being identified and extracted from the chromosome using various enzymes. These enzymes will be particularly restriction endonucleases. They can be EcoR1, BAMH1, Hint3 and many more. The required gene is identified means which trait needs to be incorporated into the host organism. Depending on that, the required gene will be selected. Then in the second step, the plasmid is extracted from the bacterial cell. Again, the steps involved for plasmid extraction will be similar to that of DNA extraction, only the last steps will be different, or here in the DNA we select the isolated gene, and the last step in the plasmid extraction will be we are taking the whole supernatant for further processing. Whereas, in the third step an enzyme is used to cut open the plasmid, that is for creation of a nick, for creation of a site for the insertion of the transgene. This nick is being created, and we have to keep in mind that is for both the isolation of gene of interest as well as for creating a nick, the same restriction endonuclease is used. This is why, because it will create the complementary sites similar for both as well as in plasmid as well as in the gene of interest. Then the next step is the required gene is inserted into the plasmid and sealed by an enzyme, that is there can be gap when this insert is being joined with the help of the enzyme ligase. So, this gap needs to again be filled by using several other enzymes. Then the modified enzyme is inserted into the bacterial cell and it produces a GM, that is genetically modified organism. It can be a GM crop or an GM animal. This modified plasmid basically contains the plasmid which has been isolated as well as the gene of interest which is isolated from any organism, and this gene of interest is naturally not present in the host organism. Then this bacteria will reproduce rapidly, producing new bacteria with required traits. Thereafter, the major challenge lies in selection of the recombinants, which is basically done with the help of blue white colony selection, with the help of many detection analysis, as well as with the help of presence of any selectable marker in the vector molecule. It can be an antibiotic resistant marker, that is it can be ampicillin resistance, kanamycin resistance, neomycin resistance. So, what will happen when we are growing a mixture of cells in a culture which contains both the transformant as well as non-transformant in presence of an antibiotic containing media, the transformant will contain antibiotic resistant, they will survive, they will multiply into the media, whereas the non-transformant being not containing the antibiotic resistant marker, they will be sensitive to these antibiotics and they will die, they will not be able to grow in this particular media. So, based on that, in the end the transformants needs to be selected, and thereafter these transformed cells needs to be expressed either in vitro as well as in vivo. The in vivo expression is basically done with the help of bioreactors, whereas these bioreactors are live cells, whereas the in vitro is done either in test tubes or in presence of suspension culture in the media containing bioreactors. In the next lecture, we are going to learn principles of cloning and applications of cloning. Clones are basically the exact copies which are produced asexually, and this cloning technique is very important to produce large number of copies of cells which contains transformant as well as non-transformant in a media. So, thank you for joining us on this enlightening journey through the steps of genetic engineering. I hope this video lecture has deepened your understanding and sparked your curiosity to further explore this fascinating field.